So, good morning or good afternoon, everybody. It's 9 a.m. at the East Coast from the perspective of our host, or 6 p.m. in Europe from our perspective. It's a great honor and a pleasure to introduce and welcome another highly distinguished speaker in our seminar series, Marc Bajenoff. Marc started his career in biochemistry. He obtained a PhD in immunology. He was in France, in Marseille. He was then a postdoctoral fellow of Ron Germain and Bethesda at the NIH. Afterwards, he returned to France and he joined the laboratory of Nicolas Gleichenhaus in Valbonne. And afterwards, he became a group leader at the Centre d'Immunologie de Marseille-Lugny. And he is now a research director at the Centre National de la Recherche Scientifique. I hope I got this kind of okay in French.
Yes, perfect.
And he's a distinguished researcher. He has won numerous awards from the National Academy of Medicine in France. He got an ERC Consolidator grant, which is the most prestigious award and grant you can get in Europe for mid-level, mid-career scientists. He has a great number of publications. He has an outstanding record of achievements. He is an imaging specialist. He fallen in love with imaging and with stroma cells. He did great contributions to our understanding how stroma cells interact and impact the differentiation and migration of B and T cells in lymph nodes. He started his work together with Ron Germain, and ever since he continued along those lines. He has made outstanding contributions to our understanding of the dynamics and the function of stroma cells, not only in lymph nodes, but also in the thymus and in the skin. He has done elegant fate mapping studies related to Langerhans cell development, follicular dendritic cells, even mast cells or epidermal dendritic cells. And he has also done work on understanding of remodeling of vasculature in lymph nodes. And I assume we're going to hear some very nice work along those lines. And I'm expecting to see a firework in pictures and imaging. You told me, or a thunderstorm, you now told me you are threatened right now by a thunderstorm in Marseille. So I hope we're going to get smoothly through this meeting. But I'd like to finish my introduction with saying it's an absolute pleasure to have you here. Thanks for joining us. And as usual, I like to start, or we like to start with a career-related question. So normally we're asking people, what are your key advices to young scientists or people who want to start a scientific career? Now we've discussed a little bit before, and we have decided, I also would like to ask you, what are sort of big things where you say, don't do this at the beginning of your career? And with that, I would like to give the word to you, and thanks again for coming.
So thanks a lot, Dietmar, for the kind introduction. Well, regarding your question, I would like to say to young scientists, maybe try to combine as many techniques as you can. I will just briefly tell you a story that happened to me during my PhD. I was studying naive T cell activation in lymph nodes. So we were using TCR transgenic adoptive transfer experiment, and we're using flow cytometry to monitor their activation in lymph node. And we found out that actually we couldn't get these cells, we couldn't see them. And then we thought that maybe, well, they were activated in the skin because we injected the antigen in the skin. And we said, wow, that's nice because it will be a paradigm shift, blah, blah, blah. And then someone in the institute was doing imaging. And then he told me, well, why don't you look with your microscope? And then I used this microscope and I said, oh, but they are there. It's just that I cannot get them out of the tissue. And then I said, okay, so now I will never use only flow cytometry, but I will combine techniques. And that's how I got into imaging in the first place. And then stroma came after that. So please always combine multiple techniques to address your question. That would be one of my advice.
Oh, perfect. I think that's a very, very good and very important advice. And with that, I'll give the word to you. And
okay, so then I will share my screen. So does it work? Do you see my full screen?
Yes.
Yes? Okay. So today I would like to talk about stromal cells and how I believe they represent a very strong promoter of macrophage homeostasis and function. But before going into more specific question, I would like just to give a feedback for the younger ones, because as I mentioned during my early career, I was really interested in lymph node immune responses. And I got interested in stromal cells. So we knew that in a lymph node, you have T and B cells, but we also found out actually you also have some stromal elements that usually people and immunologists tend to overlook. But now this view has changed. And actually, when you use an electron microscope, you can see these immune cells, which would be these round cells, but you can also appreciate some weird structure. Here we have some lymphatics. You know also that you have iron endothelial venule, and you also have here some fibroblastic reticular cells. And I got interested specifically in those cells. And it turned out that we found out that these cells form a 3D meshwork on which actually T and B cell migrate. And on top of that, this reticular meshwork feed these T and B cells. So you can consider them as a kind of a spider web. And the T cells would be these little lymphocytes crawling on the fibers of the spider web. And somehow these fibroblasts can be considered as niches for lymphoid cells. So in 2015, we said, well, maybe this fibroblast or this stromal element can also control other immune cells, perhaps in lymphoid organ, but also outside the immune system. So how about macrophages? And then we started to understand if actually the stromal cells could act as niches for macrophages in different tissue. And at that time, there was also a very nice paper from Rosa and Medzitov describing this so-called two-cell circuit, stating that stromal cells can feed the macrophage and that in return, the macrophage can provide somehow a feedback to their niche, creating this virtuous cell circuits. And then we were asking, can this unit function actually have some biological relevance? And can this regulate key processes? So we all know that macrophages exert key biological functions. So I don't know how many times you have seen this slide that I like a lot as well. But macrophages, as you know, they have immune function, that's a given, but they also have some trophic function. And they are probably important in brain development. They control bone homeostasis. They control iron homeostasis in the spleen. So there is no doubt that macrophages are important. And I think for me, the best example about this function of macrophages can be observed in CSF1 knockout mice. So CSF1 is a very important growth factor for most macrophages. And you can consider that with few exceptions, CSF1 knockout mouse, they lack tissue-resident macrophages. And the result of that is that actually the mice suffer from many different problems. They are dwarf, they are deaf, osteopetrotic, infertile, and on the B6 background, they will eventually die around 4 to 5 weeks of age. So clearly macrophages have very important functions. So how can we reconcile that with the stromal system? Well, the model right now, and maybe it's a dogma, but that could be discussed, is that the reason why these mice are suffering so much is because you have multi-organ failure, because the niche cells are doing actually key function. But in order to do that, they need macrophages. So that's why they would produce CSF1, so that the macrophages will come close to them, and then the macrophages will provide this feedback system to the niche cell. So now the niche cell can do its normal function. But if you remove CSF1, then you remove the macrophages, you would remove the feedback system, and then the key function cannot be done, cannot be performed by this niche cell anymore. And then that could somehow explain why these CSF1 knockout mice have so many problems. So it's very important to understand what these macrophages are doing in every single tissue to understand all these phenotypes. But how can we do that? How can we specifically address the function of macrophages? So here I have listed some commonly used approach to study the macrophages. I think that right now the most commonly used technique that I can see in the recent paper is the use of CSF1 or CSF1 receptor blocking antibody. Because when you do that, when you treat mice with this antibody, then you will directly induce the deletion of these macrophages. The problem with that is that it's only a transient deletion, because the monocyte at the end of the treatment will come back and give rise to other macrophages. And it's also non-specific, because you will kill all the macrophages of the mouse when you do that. So the main issue right now with this is probably the transient effect, because if a phenotype needs weeks or months to develop. There is no way you can see that with this transient deletion. So there are other techniques to delete macrophages on top of this antibody treatment. You can also use genetics, and for example, to have these macrophages express diphtheria toxin receptor, and then you treat with the toxin, and then you delete the macrophages. So it works, but then you have to consider what is happening in your mouse, because when you do that, you kill the macrophages. So this will trigger a massive cell death and an associated massive inflammation. The problem is that you just kill the macrophages that are supposed to get rid of that inflammation. So you will probably sustain the inflammation. So if now you see a phenotype in your mouse, is it due because of the loss of macrophages, or is it because of the inflammation that you trigger? So one way of doing that would be to create more specific CRE macrophages, maybe with DTA system too, but then you would have some problem as well during the development. But anyway, I think that the paucity of CRE-specific macrophages is slightly improving. But the main issue to do that is that for each specific macrophage that you want to study, you should create a mouse that would specifically target that macrophages. And it's not that easy. It requires a lot of time, money, and I think a lot of luck, because to my knowledge, there are not that many mice that are very specific. So in the lab, we have been asking, how can we actually try to find another way to target macrophages, maybe by taking advantage of the stroma niche and macrophage interaction. So let me introduce the concept of the macrophage niche that I've slightly introduced. So we know that in most tissue, macrophages are immotile and they are usually long-lived. So we can debate about why they are immotile. They are non-mutually exclusive explanation to that. So maybe they occupy each their own territory. So because they know exactly where their neighbors are, so they basically are constrained in their capacity to move. So that's why they stay there. Or there is also another explanation that there is a niche that is taking care of them. And then this niche would have several functions. The first would be to anchor the macrophage. So that would explain why the macrophage is not moving around. The second is that this niche should also provide growth factor to this macrophage, for example, such as CSF1 or such as IL-34 if you are longer on cells, or some microglia cells. And a third function that I think is quite important for the niche would be to instruct the monocytes or the pre-macrophages to become tissue-resident macrophages. If you think about that in the adult, the monocytes in the blood, they can end up in any different tissue, right? So that if they go into the liver, they can become a Kupffer cells. But if they end up in the lung, for example, they can become an alveolar macrophages. So locally, they have to be instructed. And probably this is one of the functions of the niche. So how can we take advantage of that again? Well, our idea is very simple. First, let's consider ancient civilization of human beings. We all know that humans need water, and that water geography at that time used to control human distribution. For example, in Egypt, if you look at all the big cities, they used to develop around either the Nile or the Red Sea because they needed access to water. If we assume that macrophage needs CSF1 just like humans need water, and that CSF1 location would control the distribution of macrophages, then you can end up in any organ with maybe different niches that produce CSF1. But in that case, for example, the niche 1 would produce CSF1 for the blue macrophages, while the niche 2 here in purple would produce again CSF1, but for a second type of macrophages. So our assumption and working hypothesis would be that if we dry up one source of CSF1 by targeting CSF1 in specifically one niche, then we should permanently remove nearby macrophages. And when I say permanently, it's because when CSF1 is gone, then it's a no man's land for macrophages. And now we have as much time as we want to study the phenotype, because the monocytes can never replenish that niche. So now the game becomes, what is the identity of this macrophage niche? And can we actually alter them by deleting CSF1 in those? So over the recent years, there are several papers that have described niches and how actually stromal and structural cells can actually act as niches. For example, in the lung, you have epithelial cells that can produce CSF2. In cavity macrophages and in the spleen, for the red pulse macrophages, it seems that now we have fibroblasts that can secrete either RLDH or CSF1 to control respectively their actually macrophages. In the lymph node, in the subcapsular sinus, now it's not a fibroblast anymore, but it seems that it can be a lymphatic endothelial cells. Okay, and in the liver, things can get even more complicated because now it's the cooperation of several cell types that can actually teach the Kupffer cells what they have to do. So today my talk will be divided in two separate parts. I will tell you a little bit of a story that we have right now, which is about a niche for blood monocytes. And the second one is more, I would describe a new strategy to study stromal cells in vivo. So let's start with the niche for blood monocytes. And why did we decide to study that? Well, I told you that usually monocytes, they are in the bone and they live as classical monocytes. And when they are there, they have two choices. They can either become tissue-resident macrophages. And I told you that most likely, I would favor an hypothesis in which there is a niche that will be there to feed them. But these classical monocytes can also give rise to intermediate and then to non-classical monocytes, also called patrolling monocytes, because they patrol the surface of the endothelium. And then we were wondering, is there a niche as well for these monocytes? Because actually they are not immotile, they keep moving. So how can actually the niche concept could apply to these monocytes that are continuously moving? And when we started that study, actually, it was assumed that the endothelial cells would represent the niche for this monocyte, simply because it would be very simple for them, because of their location, to produce CSF1 in the bloodstream. And then the monocyte that would just pass by, then they will just consume this CSF1. Okay. So we decided to test that hypothesis. And first, what we did is that we wanted to delete CSF1 in endothelial cells. In order to do that, we used Ty2-Cre CSF1 flux mice, and we analyzed the blood monocytes based on the regular gating strategy, in which you have LY6C and CD43. You have the classical monocytes here that can give rise to intermediate monocytes that will eventually become non-classical monocytes in there. So when we looked at the blood of these Ty2-Cre CSF1 flux mice, we noticed that in the control, you can see the 3 population, but that in the Ty2-Cre in the mutant mice, we are missing 1 population, which are the non-classical monocytes, and intermediate ones were actually affected as well. So we said, okay, fine. So, but Ty2-Cre, it's a constitutive Cre, so we are deleting CSF1 not only in the endothelium, but also in all the hematopoietic system because of lineage tracing. So let's try to delete it only in the adult mice. So in order to do that, we use the CDH5 Cre82 CSF1 flux mice. We treated adult mice with tamoxifen, and we basically observed the same phenotype. So there is a paucity of non-classical monocytes in the blood of these mice. So CDH5, okay. So here are the numbers in which you have the Ty2-Cre and the CDH5. In red, you would have the mutant mice, and in white, the control. And you can see that in terms of numbers, the classical monocytes do not seem to actually be altered, but the intermediate and non-classical monocytes are basically gone in the blood of these mice. So CDH5 and Ty2 also target the lymphatic endothelial cells. And as we have shown that lymphatic endothelial cells can promote the survival of some type of macrophages in tissue, we decided to see if they would also control the survival of non-classical monocytes in the blood. So for that reason, we use PROX1 Cre32 CSF1 flux mice to delete CSF1 only in lymphatics, but we didn't see any defect. So it's purely a vascular endothelium defect. Okay, so these monocytes are gone from the blood, but is there a problem with the macrophages then in tissue? So we had a look in the Ty2-Cre CSF1 flux mice, and we had a look at a bunch of different organs, and actually we didn't see any problem in the macrophages of these mice. Fine. But if these monocytes that are absent from the blood, then maybe they are now in the tissue for whatever reason. So we also checked for the presence of these monocytes in various organs, and we didn't see that. Basically, the monocytes that are also in these organs, they are also gone. So it's a general phenotype of this monocyte disappearing. So then we ask, what could be wrong? Why are these monocytes gone in those mice? So we assume that there should be maybe 2 main reasons. The first one is that there could be a problem in the bone marrow with monopoiesis, but we assume that it should not be the case because in the end, the classical monocytes in the blood are perfectly fine. So it wouldn't make any sense that there is a problem here, or at least it should not explain what we observe. So we assume that maybe it would be a problem of survival of these non-classical monocytes in the blood, because CSF1 is mainly known as a growth factor for myeloid cells. So we tested that by doing this very first simple experiment. We took different hosts, Ty2-Cre CSF1 flux mice and control mice. And then we injected in their bloodstream the blood that we just took from ubiquitin TD tomato mice, in which all the cells that are traveling in the blood at that time would be red. And then we follow the fate of this monocyte that we grafted in the two types of host. As you can see in gray here, the monocyte, the non-classical monocyte that have been transferred can survive up to 5 days in the blood of these mice, and then they will die, which is basically the lifespan of the non-classical monocytes. But when these monocytes are grafted in a Type 3 CSF1 flux mice, their survival is actually way lower. And actually, after 2 days, they are basically gone in the blood of these mice. So we also did the same experiment, but now we transferred only non-classical monocytes isolated from NR1A GFP mice. And we looked at 2 days. And again, we saw that actually in the Type 3 CSF1 flux mice, these monocytes were gone. Okay, so we assume that most likely people were right that the endothelial cells produce CSF1 in the bloodstream and that the monocytes are actually feeding on this CSF1. And then we measure the level of circulating CSF1 in the serum of these mice. And then we got a surprise because actually you can see here in the Type 3 or CDH5 CRE-RT2 CSF1 flux mice, there is no difference. So CSF1 is still produced in the blood of this mouse. We were using ubiquitin CRE-RT2 as a positive control. So it means that in those mice, the monocytes are gone, the non-classical monocytes are gone, despite the fact that the circulating level of CSF1 is absolutely fine. So probably what are these monocytes looking for is CSF1 not circulating, but probably attached somehow to the endothelium. So we came up with this metaphor, which is an animal metaphor, where we would like to compare non-classical patrolling monocytes to cow that keep grazing on the surface of the endothelium, because in the end, what they want and what they are looking for is CSF1 that is produced by the endothelium. But of course, monocytes are not happily grazing, right? They are constantly enduring shear stress. So we were assuming that if they would adhere and migrate to scavenge CSF1, then there should be molecules that would allow them to do that. So we know that integrins are important, for example, such as LFA1. But we also know that, for example, for T cells to enter in lymph node, chemokines are also important. And they are an important part of the equation. So we said, okay, so what chemokine could be actually acting and promoting the scavenging of CSF1 on the surface of the endothelium? And we got interested in CYP3CL1. So why? Well, first, CYP3CL1, so its receptor, is expressed by all monocytes. So fine. And then CYP3CL1 is a very interesting chemokine because unlike the vast majority of the other chemokine, it's produced as a transmembrane chemokine, not as a soluble one. So if the endothelium would produce CYP3CL1, then maybe the monocyte can use it to adhere to the endothelium and then to scavenge CSF1. So we wanted to really look at that. So people have reported that the endothelial cell can produce CYP3CL1, but it was always in vitro or using activated endothelium. So we really wanted to do that in the real mouse to see if that would be the case. But first, we asked the question, is the endothelium actually really producing CYP3CL1? So we isolated blood endothelial cells from different tissue, and we measure the level of expression of CYP3CL1. And as you can see here, it's variable, but it seems that almost all the endothelial cells are able to produce CYP3CL1 at steady state, with one exception, which is the liver. And just keep that in mind because I will come to that at the end of my talk. So then we said, okay, but in order to prove that CYP3CL1 is important, we need genetic tools. So we were lucky enough to collaborate with David Dombrovitz in France as well, who just has made a mouse, which is both a reporter and a conditional allele of CYP3CL1. So here you have the construct. So if you don't have any CRE expression, any cell that expresses CYP3CL1 will express mCherry. So this is the reporter part. But as soon as you cross this mouse with a CRE line, then any cell that expresses CRE will actually delete the third exon and mCherry as well. So now you will delete CYP3CL1 expression and mCherry. So first we had a look at this mouse and just at the flux mCherry mouse. So without any CRE, so we looked at different bunch of organs, and here I'm just showing you 3 organs. So if you look in the kidney, in the glomerulus, this is here CD31 staining in green, and you can see here in white TD tomato expression, so mCherry. And you can see that actually the endothelium here is expressing mCherry. So it's expressing CYP3CL1, and you also have here some fibroblasts that express CYP3CL1. Now, when we cross this mouse to the Ty2-Cre mouse, then you can see that there is the disappearance of the hemcherry staining in the endothelium, but we maintain it in the fibroblast. So now we have a mouse in which only endothelial cells are missing CYP3CL1. In the lung, it's basically the same with endothelial cells, capillaries expressing CYP3CL1. We also have some expression here in the epithelial cells. And when we cross this mouse with Ty2-Cre, then we lose expression of CYP3CL1 in the endothelium, but not in the epithelium. As for the liver, actually, as we saw when we quantify the level of CYP3CL1 expression, there is no expression in the sinusoids, and actually it doesn't change when we delete it with Ty2-Cre. So what is the consequence? So is there anything wrong with these mice when we delete CYP3CL1 in endothelial cells? So here is the blood phenotype of these mice, specifically on monocytes. And as you can see here in green, you have the mutant mice, and you can see that there is a paucity of non-classical monocytes in the blood of these mice, whereas the classical monocytes are not affected. So the phenotype is not as strong as when we delete CSF1, but as a comparison, we looked at the CYP3CL1 knockout mice, in which now all the cells of the mouse do not have the receptor. And if you look here, actually, the phenotype is almost comparable, as if deleting CYP3CL1 only in endothelial cells is sufficient to phenocopy what we observe in terms of monocytes, of course, in the CYP3CL1 full knockout mice. So we came up with this model that maybe the monocytes actually first engage CYP3CL1 on the endothelium, and then they will use that to adhere to the endothelium and then to scavenge CSF1. So we wanted then to formally prove that CYP3CL1 would facilitate the uptake of CSF1 by monocytes. So how did we do that? Well, we know that when CSF1 binds to its receptor, so CSF1 receptor, it's internalized and then degraded, meaning that if you have a lot of CSF1 that is around, then you will have a lot of internalization of CSF1 receptor, and you can use actually the surface expression of CSF receptor 1 as a proxy for the amount of CSF1 that the cell has been able to engage and interact with. And as an example here, if you take a regular white-type mouse, and if you inject CSF1 in the blood of these mice, and if you look at the receptor for CSF1 on the surface of classical, intermediate, and non-classical monocytes, you can appreciate that actually the receptor is downregulated when you inject CSF1. And then actually here you can quantify, and all type of monocytes downregulate CSF1 receptor when they bind CSF1. So here is the experiment that we did to actually test if CYP3CL1 would allow the cells to actually engage CSF1. We relied on shield chimeric mice, in which we irradiated only the rear part of the mouse just to make some space in the bone marrow of the rear legs of these mice. So we had two types of different hosts, the white-type recipient and the CYP3CL1 knockout recipient. And we grafted ubiquitin TD tomato bone marrow in these two different hosts. So in the first one, the red cells, the fluorescent one that come from the donor, will express CYP3CL1, and they will end up in a mouse in which also the monocytes express CYP3CL1. So they won't. You should not expect any competitive advantage because the two kinds of monocytes have the same amount of receptor on the surface. Whereas in the second host, the host monocytes don't have any CYP3CL1 on their surface. So you would expect the donor cells to express, of course, the receptor, and then because of that advantage to engage CSF1, and as a result, to have less CSF1 receptor on their surface, telling us that actually they have engaged CSF1, and that's why it's internalized. So we have measured the level of CSF1 receptor expression on the surface of these donor monocytes. And as you can see here, you have the white-type situation. You don't have any, it's basically the control. And you can see that the receptor is downregulated in the white-type cells, but only when they are in competition with CYP3CL1 knockout monocytes, meaning that they have a competitive advantage to actually scavenge CSF1. And actually, I'm not showing you here, but they have also, of course, a competitive advantage of survival. Okay, so it seems that endothelial cells are the niche for monocytes, but then came the question, what are the endothelial cells that are important for providing CSF1 to these non-classical monocytes? And last year, there was a very nice paper that mainly focused their attention on the bone marrow and at the niches of the non-classical monocytes in the bone marrow. And they concluded that the bone marrow sinusoids were the niche of the non-classical monocytes in the bone marrow. And they also suggested that maybe there would be also the niche for all the non-classical monocytes that are in the bloodstream. But the problem is, how do you demonstrate that? And actually, I think that there are two exclusively model, for example, this time, in which you would have the model one would say that maybe, yes, the bone marrow sinusoids are the exclusive niche of all the non-classical monocytes. But then if it was the case, you would say, well, the number of niches is super low because this is only the sinusoidal cells in the bone marrow. As a result, because there are so few niches, you would expect each of them to produce huge amount of CSF1 to feed all these non-classical monocytes that are traveling in the blood. And obviously, the access to the bone marrow for these monocytes that are traveling in the blood should be mandatory because that's how they would be fed. The second model would be a different model in which now all the endothelial cells of the vascular tree would act as niches. So now the number of niches would be very high because every endothelial cells could be actually a niche. Then the production of CSF1 by sinusoids would become almost irrelevant if that would be the case. And the access to the bone marrow to be fed would also be dispensable because the non-classical monocytes in the bloodstream would not have to go back to the bone marrow in order to be fed. So how can we actually distinguish these two models? Well, if you remember, I told you that the liver endothelial cells do not produce 6'3"CL1. And I also show you that actually 6'3"CL1 is probably important. But we said, wait, the liver endothelial cells are very specific because they are sinusoidal. And here in the bone marrow, we're also dealing with sinusoidal cells. So what if actually the bone marrow sinusoidal cells do not express 6'3"CL1? And can we take advantage of that in order to make a mouse in which we could delete CSF1 in all the endothelial cells, but not in the sinusoidal endothelial cells? So we decided to try it. And we made a 6'3"CL1 Cre-ERT2 mouse that would, in our head, target all the endothelial cells, but not the one in the bone marrow. So as a proof of principle, first, we crossed this mice with Rosa TD tomato. And here you have a few organs in which you can appreciate the recombination in the brain, the lung, and the heart endothelial cells. You have a pretty good recombination. But in the bone, actually, there is very little recombination, which would fit with what we observed. Then when we imaged the bone marrow of these mice, actually, we could see a lot of red cells, but actually these cells were not endothelial cells. They were mostly fibroblasts and pericytes that were lining along the sinusoids. And the only few sinusoidal cells that we can see would be here in this screen. So most of them are not labeled. So we assume that, okay, so now we could use this mouse, cross it with CSF1 flux mouse, and see what happens. So that's what we did. We generated 6'3"CL1 Cre-ERT2 by CSF1 flux mouse. And here are the results for the blood. And what we noticed is that we also observe a paucity of non-classical monocytes in the blood of these mice. So probably promoting the idea that all the endothelial cells can actually produce CSF1 and that the sinusoidal cells might not be the one that drive the survival of the blood circulating monocytes. When we looked at the blood, at the bone marrow of these mice, actually, then we didn't see any paucity of non-classical monocytes. So probably the bone marrow sinusoids are indeed providing CSF1 locally to the monocytes that are in the bone marrow, but they are probably not the one in charge of feeding these blood circulating monocytes. So I also told you that if bone marrow sinusoids would feed all the non-classical monocytes, we would expect them to produce huge amount of CSF1 because even the circulating level of CSF1 are not sufficient to sustain the monocytes in the blood. So then we just simply looked at the production of CSF1 by measuring the transcript of CSF1 in different types of endothelial cells. And as you can see here, all endothelial cells seems to produce CSF1, and the bone is not particularly producing a lot of CSF1, which would be a prerequisite for the first model. Then we asked, okay, so there are several types of endothelial cells, right? They could be artery, capillary, vein. So is there any one subset that is making CSF1? So for that, we took advantage of an atlas paper in which they actually sequence all the endothelial cells of different organs, and then they did cluster them in different subtype of endothelial cells. And then we simply ask which cell produce CSF1. And if you look here, actually in different type of organs, well, actually all type of endothelial cells seems to produce CSF1. So we don't think that there is a specific subset of endothelial cell that is producing CSF1. So finally, I told you that actually access to the bone marrow would be very important for the first model, because they would need to access to the bone marrow to be fed. So we wanted to test that hypothesis by playing with actually CXCL12, because CXCL12 in the bone marrow is a very important molecule to maintain monocytes in the bone marrow. It acts as a retention signal, whereas CCL2 is acting as a leaving signal. Let's put it that way. So we know that CXCL12 is expressed by fibroblasts in the bone marrow, and we wanted to actually delete CXCL12 in order to force the monocyte to get in the blood, and then ideally they should never be able to come back to the bone marrow, and then we can test what happens. So we wanted to do that in an inducible system, so we didn't want to use a constitutive Cree because we didn't want to mess up the development of the mouse. So in order to do that, we said, okay, let's make a new mouse in which we can conditionally delete CSF1 in adult fibroblasts. In order to do that, we decided to generate a biglycan Cree-ERT2 mouse, simply because biglycan is expressed abundantly by almost all macrophages in the body. So we generated first a BGN Cree-ERT2 by Rosa TD tomato, just to check if actually this would work in the bone marrow. And here is the bone marrow of this mouse, where you can see that actually these cells are actually fibroblasts that have recombined. So, okay, so now we generated BGN Cree-ERT2 CXCL12 flux mice. And this is what I'm showing you here first in the bone of this mice. And as you can see, this is a very drastic phenotype. All monocytes are almost gone in the bone marrow of these mice. Okay, so now what happened in their blood, right? And actually, if you look at classical, intermediate, and non-classical monocytes, they are all perfectly fine. Then their number, when opposed to control mice, are fine. They are normal. And we even have more non-classical monocytes in the blood of these mice. So maybe because they cannot extravasate, that we cannot really explain. But what is clear from that is that the access of the bone marrow is not mandatory to maintain the non-classical monocytes that are in the blood. So the model that we would like to promote is that the peripheral endothelial cells can act as niches for non-classical monocytes. They can do that by providing adhesion molecule for non-classical monocytes. They can also do that by providing CSF1 for the survival of the non-classical monocytes. And actually, it has been shown that they also produce DLL1 as a transmembrane molecule that is mandatory for the transition of the intermediate to non-classical monocytes. Okay, so if you have this picture in mind, then you might ask yourself, well, actually, if classical monocytes that are in the bloodstream constantly engage all the molecules that I've just talked about, why do we have only 40% of the non-classical monocytes in the blood? And it's even 10 in human. I mean, why not 90 or 100%, right? They should all convert into non-classical monocytes if all these molecules are accessible. Okay, so some classical monocytes just get out from the bone marrow, so it can't be 100%, but it should be higher than 40% and definitely more than 10%. Well, there is something that was missing in my drawing. It's the glycocalyx. Because people tend to forget that every cell is surrounded by a glycocalyx. And endothelial cells can have a very thick glycocalyx, up to 5 microns for large vessels. And this glycocalyx has a unique function. And the main one is to prevent any adhesion from cell that is circulating in the blood. You don't want your platelets, your monocytes, your T cells, your neutrophils to adhere to the endothelium, right? So that's the main function of the glycocalyx. The glycocalyx is even charged negatively. And every cell that is in the blood has also its own glycocalyx, which is negatively charged. So you have a double system to actually prevent that, negative charge and then physical constraint. The main issue is that all the molecules that are important for the survival and transition of these non-classical monocytes are transmembrane molecules, ICAM, DLL1, CXCL1. They're all transmembrane molecules. CSF1, we can debate if it's inside the glycocalyx or on the surface, but you get the point, right? And then the question is reversed. Then why not 10? Why not 0? Because actually, if the classical monocytes can never access to the molecules that are important for their differentiation into non-classical and their survival, there shouldn't be any non-classical monocyte to start with. So what's going wrong? I don't have a definitive answer because what I'm about to tell you is very difficult to prove, but this is my model, let's put it that way. I believe that at steady state, all these molecules are inaccessible. So monocytes do not engage any of these molecules. So in theory, they should not be able to become non-classical and they should not be able to survive. But there is something that is very specific to the glycocalyx, is that it will shed immediately, even if there is the slightest injury. And when I'm talking about injury, I'm not thinking about bacteria or viruses. Even shear stress is able to induce daily injuries. So what I'm thinking is that whenever you have a little bit of shedding as a steady state condition, then 6'3"CL1 will become visible. And now the monocytes will actually be able to use that to adhere, and then they will look for CSF1. And this is how the system would work. And of course, if you have a bacterium or a virus that actually activated, trigger this shedding, then the monocyte will actually capture the bacteria and the pathogen. But the primary aim of this monocyte is to capture its trophic factor. So that's the model that actually we are favoring. So now I would like to describe a new strategy to study stromal cells. But first, why did we have to design the new system? That's what I will try to explain you. So I told you that I like this comparison a lot with humans that need water and macrophages that need CSF1. And I told you that as part of this cell circuitry that Ruslan Medzitov promoted, it said that fibroblasts or niches will produce CSF1, for example, and that macrophage in return provide a feedback signal. So in vitro, it has been shown that fibroblasts produce CSF1, macrophages respond to that, and they will produce PDGF, which is a growth factor for fibroblasts. And in the end, you would have somehow an equilibrium between the two populations. Okay, so now if we compare that to what I've told you with water and human, well, the river would produce CSF1 water to human beings, but there is no feedback in there, right? Because the river is not expecting something back from human beings. So the model is kind of, okay, not working that well in terms of cell circuitry. So can we think about another more natural model? And I think that this model is pretty good. Let's assume that the macrophages is this bird, and this bird is looking for food, and the food is actually the seeds, which I would compare to membrane-bound CSF1 produced by the niche, which would be the tree. Okay, so now if you think about the cell circuitry, the question to ask is the tree expecting something back from the bird? So some people would say, yes, obviously. I mean, the tree will spread the seeds, and then it will help the tree at the population level. Yes, but it's not exactly what this system means. In this system, the niche is producing CSF1 as a membrane-bound, usually factor, and then the macrophage will come and produce a direct feedback that the niche will use. But in that case, the tree is not benefiting directly from the bird, right? So we said, is there a way to study if there is a real feedback in vivo and not in vitro? So how can we assess this if this feedback is existing? And I'm telling you right now, I don't have the answer to that. What I'm just about to show you is the approach that we are developing to get this answer. So if you take a CSF1 wild-type mouse, and then you take our metaphor of birds and trees, then actually the trees produce fruits and the macrophages will consume that. And then you expect to have a feedback. Fine. If you take a full CSF1 knockout mouse, then the CSF1 is not there anymore, the macrophages are not there anymore, and you can think, yes, but now you can ask if there is a feedback. But I don't think that the CSF1 knockout mouse is a good model, because the model is so messed up. Remember that the mice die around 4 to 5 weeks of age. So these mice have way more problems than just asking if there is a feedback or not. So we need something more subtle. The problem is that if you take a CSF1 knockout wild-type mouse, then now all the niches produce half of what they should do. So the macrophages would have to adapt to that, but this is not the ideal situation, because what you would ideally need is a system in which part of the niches cannot make CSF1 and part can. And this is actually what I represented here. The ideal situation would be a model in which half of the niche could produce CSF1 and half could not produce CSF1. And now in the same organ, you can ask, is there a net benefit from the niche that produce CSF1? Do they survive more, for example? So how can we do that? Well, an old system to do that would be to use aggregation camera. So you took embryo cells, you mix them, then you generate a mosaic offspring, and then you can ask exactly this type of question. The problem is that it's very tedious, and the mosaicism would be highly variable. Some mice will have a very low mosaicism, some mice will have a super high mosaicism. And the main problem is that it's non-inheritable. You cannot transmit this mosaicism using that system. So we said, how can we design a system in which we can get what we want in terms of fibroblasts and niches? And then we thought about the X chromosome, because naturally, every female actually has developed a way to ensure that it will have the same amount of X gen product between as a male, right? Because male have X and Y chromosome and female have twice that number. So they need to inactivate one X chromosome. So how does it work? Very early during embryogenesis, every female cell will have to make a choice, which is to inactivate either the paternal X chromosome or the maternal X chromosome. And they do that. And then this is fixed. And this is fixed. And because it's happening very early in the embryo, this choice will be transmitted to their progeny. So in the end, what you end up with is that a woman is a mosaic of clones that have chosen the maternal or the paternal X chromosome. And one example of that is the calico cat with these colors. So you know that it's the color code is encoded in the X chromosome. So the male only have one choice. So for example, if they have an orange coding chromosome, they will be fully orange. If they inherit a black chromosome, then they will be entirely black. And if you are a female, then you have multiple choices. If you inherit 2 black chromosomes, then you are black. 2 orange, you are orange. But if you inherit 1 orange and 1 black, because of this mosaicism, you will end up with clusters, because the choice, again, are made very early during embryogenesis. So how can we adapt that system to our question? Well, I told you that we have engineered a new mouse, which is a biglycan CRE-RT2. But the choice of that gene was not random. We chose specifically that gene because it's expressed on the X chromosome. And now we thought that we could play with that by generating these chimeric mice. So in order to really take advantage of that, we made even a second mouse, which is a BGN M Venus mouse. So now we put everything in the same mouse, in the same female mouse, and we even put a ROSA RFP just to report the expression of the CRE-RT2. So what would happen in this mouse? So very early during embryogenesis, you would have the 2 X chromosome inherited from the father and the mother. And then you would have random inactivation of 1 of the 2. So if the cell decides to go with the BGN M Venus, then it will give rise to plenty of different cell types. But only the cell that expresses BGN will become green. If you are a neuron and if you never express BGN, then you won't be green. So in this mice, roughly half of the fibroblasts should be expressing M Venus. The other half, on the contrary, should express CRE-RT2, but in an inducible manner. So when we will treat the mice with tamoxifen, then the fibroblasts will get CRE active, and now they will undergo the recombination. And remember the calico cat, because I think it's one of the beauty of the system. The reason why you have these patches is because the choice was made very early, and then the patches is composed of an orange cluster, and then you will have a black cluster, meaning that these fibroblasts should not be randomly scattered in the tissue, but hopefully, but unfortunately, it's not the case in all tissue, you should have clusters, island of red fibroblasts expressing CRE, island of green fibroblasts expressing M Venus, and now you can delete the gene of interest and start asking interesting questions inside the same organ, inside the same mouse. So as a proof of principle, I'm just showing you the spleen and lymph node because we like lymphoid organ. This is our baby organs, as I would say, and this is a confocal picture of spleen, here, T cell zone, and lymph node. And for example, here I have highlighted some clusters here in the red pulp. You can see here a red one, green one, even in T cell zone where you probably have TRC here, TRC in red. And in the T cell zone here, we have 2 obvious clusters. And now you can ask what would happen in those different islands. So the key question that we would like to address right now is we are bringing CSF1 flux into those mice. And we will basically ask first, is there a competitive advantage of the green CSF1 wild-type fibroblast over the red CSF1 knockout fibroblast? If there is no competitive advantage, then I would say that maybe the feedback mechanism is not happening in vivo, or we have to revise that. If there is an obvious bias towards the green fibroblasts, I would say yes, the red fibroblasts have been counter-selected. We can also ask if the CSF1 competent green fibroblasts that can produce CSF1, will they try to compensate for the inability of the red one to produce CSF1? Because maybe they talk to each other, and now there is somehow a way for the competent one to compensate for the one that cannot do that. Or maybe it will be even IL-34 that would be expressed. That we don't know. Finally, we would like to understand if the macrophage was preferentially settled in green island where CSF1 is produced, as opposed to the red islands where CSF1 is not produced. And if it is the case, then probably I would conclude that CSF1 is probably membrane bound, because otherwise it would be difficult to explain. So in the end, I hope I convince you that stroma are very important for controlling macrophages, but I think this analogy can be even refined by this picture that I like a lot. I would probably promote the idea that if you want to study macrophages, well, you could also start looking at stromal cells, because if you target stromal cells and if you control them, then you would actually be able to control macrophages. I would like also to thank all the people who did the job in my team. So Elisa Baudon, Guillaume Thierry, there was also contribution of Michel Beinen, Johanna, Merlin, Myriam Sekias. My collaborators are listed here, who provided a lot of tools and regions and mice. And finally, I will also finish by saying that right now the CML is looking for group leaders. The call is still open until June 30. So if you want to work in a very nice scientific and landscape environment, just send an email to Marc Bajenoff. And I thank you for your attention.
Well, thank you very much, Marc, for this spectacular and very inspiring presentation. I see that you have a very elegant view out of your, I guess that's your lab view. And as you all know, or I'm sure there's plenty of questions right now, but as you know, we usually make the sharp cut here and refer and hope that you're going to join Marc on Twitter, where you can ask all the questions that you have. And with this, I'd like to finish the session. Thank you very much, Marc, again for joining us. I wish you all elegant and great remaining day, and I hope to see you again next week on Global IMMUNOTALKs.
Bye-bye.
Thank you.